everyone. We will go ahead and get started in 5 minutes. Hello, everyone. Thank you all for joining today and welcome to the NOAA Omics Seminar Series. I am your host, Nicole Miller, and I am the NOAA Omics Coordinator, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the past few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted up on our Omics website. There should be a link to the website in the chat now. And as participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read these questions for the presenter to answer. So with that, our presentation today is titled Optimizing Stony Coral Tissue Loss Disease Intervention Strategies Through Whole Transcriptome Gene Expression Profiling by Dr. Michael Studivan. I'll go ahead and share my screen and give sharing permissions to Dr. Studivan and give a brief bio introduction. So, one second. Alrighty, Dr. Studivan, you should have screen sharing permissions.
Just one moment. I just need to double check that my security permissions are correct. I feel like this always happens. Anytime computers. Okay. I'm really sorry for this, Nicole. I will have to quit out of GoToMeeting and join back in, but I will be right back in just a moment. Okay?
Yeah, I'll do a quick bio.
Thanks. I'll be right back.
Dr. Studivan is a coral disease and omics PI within the AOML coral program and is interested in using omics approaches to better understand coral connectivity and resilience to stress. His research addresses stony coral tissue loss, disease transmission, and treatment, as well as assessment of coral adaptation in marginal and extreme environments. And prior to his current position at AOML, he received his PhD and completed a postdoc with Dr. Joshua Voss at the Florida Atlantic University as part of the NOAA Cooperative Institute for Ocean Exploration, Research, and Technology. So we'll give him a few more minutes to get his presentation up, and then we will go ahead and get started.
Hi, Nicole. I think you'll need to allow me to share my screen again since I popped off and came back in.
Do you have screen sharing opportunities? Oh, wait, here we go. Yes. All righty.
Excellent. I'm just pulling it up right now. Okay. Looks like it's broadcasting from my end. Can you confirm that everything looks good on your end, Nicole?
Looks great.
Great. Well, thank you so much for that introduction, and thank you all for being here. I'm happy to share the results of a project that was primarily funded by NOAA Omics, but also had support through the Florida Department of Environmental Protection and NOAA CRCP, the Coral Reef Conservation Program. This was a collaborative project led by myself and Josh through FAU's Harbor Branch Oceanographic Institute, the University of Miami CMIS, it's a NOAA Cooperative Institute based in Miami, as well as NOAA AOML. Before I jump into the specifics of the project, I want to give a quick primer on a really devastating coral disease outbreak that's been occurring since 2014 in Florida. It's a new disease called stony coral tissue loss disease that has quickly become the most devastating disease outbreak that we have ever seen in the history of coral disease research. In a very short amount of time, in a couple of years, it spread throughout all of Florida's coral reef, as you can see in the map over to the right, the red zone showing incidents of disease lesions appearing on corals. But what makes this disease so much more devastating than other observed diseases is that it affects nearly half of all the Caribbean reef building species. So at last count, it affects 22 coral reef species, and it's extremely lethal. There's really been no way to estimate just how many colonies it has affected, but it's likely in the millions now as it's spread throughout the Caribbean. Now, NOAA has taken a charge in responding to the SCTLD outbreak, primarily in Florida, but also in US territories outside of Florida. And so last year, we released the strategy for SCTLD response, an implementation plan that includes different management and research actions to improve our understanding of the disease outbreak and ways to mitigate its spread. So that report was led by Caroline McLaughlin and Dana Mendez from Florida Sea Grant and NOAA CRCP respectively, but there were other people from NOAA, including myself, that were co-authors on this. And the priorities from this response plan are, number one, to identify different modes of transmission for the disease, so how it's been spreading throughout Florida and across the Caribbean as rapidly as it has, identifying pathogens responsible for the disease, which is a notoriously difficult task for coral diseases, and then to evaluate potential treatment approaches. Now, if you're able to tune into my NOAA Omics seminar from last year, you may have seen some of the work that we've done at AOML, looking at priorities number 1 and 2, in terms of identifying transmission through sediments and ballast, as well as to attempt to identify the pathogen through some of our highly sophisticated experimental setups. But today I'm going to be talking about priority number 3, evaluating potential treatment approaches. Really, one thing has come up known as disease intervention. So historically, whenever there's been coral disease outbreaks, researchers and managers have attempted to treat individual colonies that appear infected with the disease as a way to mitigate the spread of disease among different colonies. And so one method that was successfully used for previous disease outbreaks was a mixture of chlorine and epoxy that was applied to the disease lesion itself and then allowed to cure, hoping that the chlorine would kill the pathogens. Now, when this method was used for SCTLD in Florida, it had really poor success. In fact, it was no more successful at stopping lesions than doing nothing at all. And so as a community, we realized we needed to move on to something a little bit more catered towards this disease. And so over the last 5 years, through the help of CoreRx and Ocean Alchemist, they have now developed a suitable treatment approach using amoxicillin, which is a common off-the-shelf antibiotic, and Base 2B, which is a silicone-based paste that is specifically designed to time-release amoxicillin on corals. So it was designed by this company for use in treating SCTLD. And as a result, it has a 95% success rate in halting lesions. So you treat a coral and generally that lesion disappears. Now, some work that Josh's group has done at FAU Harbor Branch, where they've continued fate tracking colonies that have been treated over time to see if antibiotic treatment is a successful means of retaining individuals in the population. And they've primarily done this with Montastraea cavernosa. It's the greater star coral that is really common all throughout Florida, and found that these colonies, of the colonies that were treated, 70% have survived after 2.5 years. And this is very minimal treatment. It's not repeated treatment. It's usually 1 or 2 times. What they found is that when you do an initial amoxicillin application on the lesions, followed by a second application within 2 months, you get the best bang for your buck. So you get the highest cessation of lesions while minimizing diver effort in the water. However, they do recognize that it might make sense to repeat treatment for high value colonies. So these are our larger colonies that are likely contributing more gametes during the reproductive phase than smaller colonies. And you can see in this photo panel down on the bottom how a coral was initially treated with amoxicillin in that second photo. It's that white band. And then 5 weeks later, you see that the lesion is much reduced in size and looks like it's starting to heal. And then after almost a year, all of that coral tissue has healed and there's no presence of any lesions. However, I do also want to mention that since 2020, Josh's group and others that have been conducting monitoring efforts in Southeast Florida have noticed that SCTLD prevalence, or the percentage of corals with lesions, has really dropped. So we're not really sure what this means as of right now, but it looks like SCTLD is moving into an endemic phase where it's still present, but in much lower abundance than it was previously. Now, there's a flip side to antibiotic intervention. Not only is it really energetically costly and expensive to treat individual corals when you're considering that there are millions of coral colonies throughout the Caribbean, there has been relatively high success here in Florida, where several strike teams have been able to treat over 10,000 diseased corals all across the Florida's coral reef since 2019, so only 4 years now, and they're primarily using amoxicillin and base 2B now that they found it to be much more effective than other methods. But on the flip side, we don't really have a strong understanding of any downstream negative effects of antibiotic treatment on corals and their symbionts. We do know that these antibiotics are found in coastal zones naturally in Florida because of human wastewater. But we don't know if treating with antibiotics might be causing some unintended consequences like antibiotic resistance. And so we really need research that addresses these kinds of gaps to see whether this Band-Aid that we're putting on the corals might impact them further down the line. And so to better understand the impacts that not only the disease, but the subsequent antibiotic treatment might be having on coral health, we conducted 2 different experiments as part of this project. The first was an ex situ transmission experiment that just looked at SCTLD transmission. And the second was an in situ intervention experiment where we treated corals in the field and then sampled them before and after. And both of these experiments, we were analyzing coral health through gene expression profiling, also known as RNA-seq. And so this is a great way to look at whole transcriptome responses to a particular treatment. Starting in with the transmission experiment, this was done in our wet lab facilities at the University of Miami CMS and NOAA OML. We have this really great custom transmission apparatus that you likely saw if you tuned into my talk last year on our efforts to identify SCTLD transmission vectors. So what makes this system really great is it separates out our water systems into independent coral jars. So we can support very high levels of replication with statistical independence. This means that any disease lesions that we see are directly the result of what we're dosing it with and not, say, a neighboring individual. We obtained fragments of 2 different coral species, Montastraea cavernosa and Orbicella faveolata. These are 2 species that are moderately susceptible to the disease. However, the fragments that we obtained through Mote Marine Lab were considered disease naive since they've been kept in land-based nurseries since before the SCTLD outbreak. We then exposed each of these fragments to strips from a larger colony of Montastraea cavernosa that had SCTLD lesions in the field. And we exposed them over a period of 2 weeks and recorded observations of disease lesions and when they occurred. From this, we also took samples for our omics. So because we had genotypic replication, we had 1 frag that was not exposed to disease and 1 frag that was exposed to disease from the same parent individual. It allowed us to look at healthy versus disease trends while controlling for coral genotype. So we had 20 individuals in each treatment per species, giving us a total of 80 samples. And when we sent this off for sequencing, we generated just shy of 2 billion reads, which is very high. I think our average number of reads per sample was something like 25 million. So really great for doing RNA-seq. Jumping into the results from this experiment, what we found is that overall differences between treatments were great. So when you compare our diseased versus our healthy individuals, there were really strong differences. This is on the order of 7,500 differentially expressed genes, and that's a lot. Generally, you consider high levels of differential expression when you have maybe 100 genes, but to have 7,500 between our diseased and healthy individuals, shown as red for diseased and green for healthy on the PCOA over to the left. Each dot here corresponds to an individual sample, and they cluster out pretty well by treatment. Now, what I want to mention is we did have some individuals that were exposed to disease but didn't exhibit disease lesions by the end of the experiment. And so these we call no active infection, or NAI. These are represented by orange. And those individuals, despite being exposed to disease, more resembled their healthy counterparts than they did to the disease counterparts. So it could give us some indication that these corals are resisting the pathogen or whatever type of disease exposure. When we take those differentially expressed genes and look at a higher level function, so if we start to piece those individual genes into gene pathways that cause some type of biological response in the corals, such as through eukaryotic orthologous groups, or COGS, that's what this figure over to the left is showing. So each row of this figure corresponds to a series of genes that have that particular function. And in this heat map, each column is showing us a pairwise comparison of treatment. So for example, our rightmost column is diseased corals versus those NAI corals, the corals exposed to disease that did not develop any lesions. And so when we have increasing intensity of the blue color, it means that we had lower expression of those gene pathways in our diseased corals versus our NAIs. And then conversely, with our increasing intensities of the color red, it means we had higher expression in disease versus NAI. And what we can see when you take a look at the broader context of this figure is that when corals are exposed to disease, it causes a large-scale depression in a metabolic and repair mechanisms. So most of these rows here, comparing our disease to our healthy in the middle column and our disease to our NAIs in the right column, most of the metabolic pathways are dark blue. So that means that they have lower expression after being exposed to disease. However, we did see an increase in expression of transport mechanisms and common stress response mechanisms. So a lot of these translation and ribosomal structure over here on the right in red, as well as nuclear structure that is very intense red, meaning that it's very much in demand in the corals expressing genes associated with that pathway. When you bring that into another type of functional annotation, in this case, gene ontology or GO terms, we have over 450 GO terms or gene pathways that were significantly enriched in response to disease, which is an enormous number. And it means that there's really a lot of transcriptional responses happening in the coral. Now, when we look at the other species in this study, Orbicella faveolata, we found a much more muted response to disease. And so I first want to mention here that we only had one individual that was exposed to disease that did not develop symptoms, that did not develop signs of disease. And so my primary analysis here, excuse me, let me back up one time. The primary analysis here is comparing just diseased to healthy individuals. So we had 575 differentially expressed genes between diseased and healthy individuals. But we do see the same kind of depression of metabolic pathways when we look at the COG patterns in diseased Orbicella faveolata, where we have a reduction in metabolic pathways, but an increase in cell transport and stress responses. We also did have 32 GO terms, or gene ontology terms, that were significantly enriched in diseased Orbicella faveolata, and 69% of those actually matched the exact same GO terms between Orbicella faveolata and Montastraea cavernosa. What's interesting here is that Orbicella was significantly more likely to develop disease lesions and in a faster time frame than Montastraea cavernosa. So it's been well established with other transmission experiments that Orbicella tends to see first lesions around 2 to 3 days, and Montastraea cavernosa is between 5 to 7 days. And so we think that this could be related to the dominant symbiont type that these corals tend to host. So there's been some great work done out of the University of Miami that's shown that different algal symbionts might have a different susceptibility hierarchy with SCTLD, where our Orbicella faveolata tend to host Durastinium from this experiment, which is more susceptible to SCTLD than Cladocopium, which is the dominant symbiont type in Montastraea cavernosa. Now, the great part about doing this experiment with 2 different species is we can look at similarities and differences in gene expression patterns across the 2 species. And so what we did here is we used this tool called OrthoFinder, which identifies genes that are the exact same in both species. And we compared our disease versus healthy individuals. So to orient you to the Venn diagram to our right here, on the far left, we have MCAV up, which in this case is comparing diseased versus healthy Montastraea cavernosa. And so these are genes that were highly expressed in diseased individuals. Then in this pinkish color is Orbicella faveolata up. Once again, genes that were highly expressed in diseased individuals. And then on the right-hand side in the 2 blue, we have genes that were less expressed in diseased individuals in Orbicella faveolata in the light blue, and then in the darker blue with Montastraea cavernosa. So what we find, of the 681 differentially expressed genes that were found in both species, the majority, or 2/3 of them, were found to be working in the same direction. That means they were highly expressed in Montastraea cavernosa diseased individuals as they were in Orbicella. And the same is true for underexpressed genes. However, about 1/3 of these genes had an inverse relationship. This means in one species, they might have been highly expressed, and then in the other species, they were less expressed, which is interesting. And when we coalesce individual genes, once again, into our functional pathways with our gene ontology terms here, we do find that overall, the similar functions are almost exactly the same between species, where we have generally more gene pathways have higher expression in diseased individuals, shown as the red dots here, with the size of the dot corresponding to the number of genes associated with that gene ontology term, and relatively few gene pathways that were underexpressed. So to put this all together, what this is telling us is that these 2 species may use the same gene mechanisms, but in different ways to ultimately modulate a similar overall molecular mechanism. So they might have adapted through time to use the same gene pathways, just in different ways from one another. And that could also explain why these species manifest lesions in different time frames. So more work needs to be done on that. It's a really cool hypothesis that came out of this study, and I really want to dive more into the molecular responses to disease in the future. Now, shifting gears a little bit. So we have a decent understanding of what happens when you expose these corals to disease in the first place. But what happens when you then treat that disease lesion with antibiotics? And so for this particular experiment, we worked within an existing long-term monitoring site in Southeast Florida. This is where we've historically done our disease intervention experiments to gauge the efficacy of dosing antibiotics. And so in this experiment, we had diseased and apparently healthy colonies that we tagged and fate tracked. And so all of the disease colonies were treated with amoxicillin and base 2B directly on the lesions, like in the photo to the bottom right. And then we came back and resampled all of our treated and healthy colonies about 2 weeks later. So we took our omic samples before and after we did that intervention, and then 2 weeks later. So we had 15 initially diseased and then treated corals, and then 19 healthy corals tracked through time, generating just about 2 billion reads across the entire experiment. So once again, we had a really high level of read depth for each sample. Just one moment here as I take a drink of water. Okay, jumping into our results here. You might notice that this PCOA shown to the left looks a lot more messy than the ones we saw in our transmission experiment. Part of this is because field environments are variable. We have different microenvironments between individual coral colonies. We can't really control for it as much as we can in the lab. So overall, we're seeing these really subtle differences between diseased, treated, and healthy individuals. But we can start to tease those apart by looking at specific pairwise comparisons. So over in the plot to the left, we have our healthy individuals once again shown in green, our diseased individuals shown in red, and then our treated shown in orange. And then the time point, either the initial time point or 2 weeks later, is indicated by open circles versus closed circles. So I don't blame you if you can't pull much from this figure. I can't really, just looking at it as a whole. So instead, we start to look at individual pairwise components, or pairwise comparisons, excuse me. So like I said, environmental variability is likely muddling the overall treatment effect. However, when we look at specific pairwise treatment comparisons, we can see that there has been a shift. So in this Venn diagram, we are comparing individual treatments and time points. So H corresponds to healthy, D diseased, T treated. Then the number that follows each letter corresponds to a time point. So 0 is time 0, and then 1 is 2 weeks later. So on the left hand of our Venn diagram here, we have healthy corals at time 1 versus healthy corals at time 0. This is just looking at overall time difference. Next, in the brown, we have our diseased corals versus our healthy corals at time 0. So we see that there's a strong level of difference. Take a look at this 321 that is in the darker brown. This is a large number of differentially expressed genes between diseased and healthy individuals. We expect this given that what we've seen in our transmission experiment. Now, when we look at our treated individuals at the second time point versus the same coral colonies before they had been treated with antibiotics, we see less number of differentially expressed genes, 181. And now what's really remarkable is that when you compare the treated individuals to the healthy individuals at the same time point, there's almost no difference. So what this is saying is that once these corals are treated with antibiotics, they look more like corresponding healthy controls than they do their original disease state. So something major is happening within coral transcription that's causing them to almost appear healthy. Obviously, we know that's not the case because they still have their lesions present after just 2 weeks following treatment, but this gives us hope that they're moving in the right direction. So when we once again look at our functional pathways with our COG groups shown in the heat map to the right, we see when we compare our diseased versus healthy individuals, we see the similar depression of metabolic pathways with some defense and stress response pathways that were elevated after exposure to disease. Now, when we compare treated versus diseased individuals and treated versus healthy individuals, we see that a lot of those same pathways are modulated to have the opposite level of expression. So that means that we're seeing higher expression rather than depressed metabolism after a coral has been treated, which sounds great. And so a big question that comes from this is whether antibiotic application is directly affecting these mechanisms, as in, can antibiotics actually change coral transcription, which is unclear, or is it some type of secondary response to shifts in microbial communities as the antibiotic is killing off pathogens? Or is it a combination of the two? We don't currently know. Here is where we get really cool. So we can do the same kind of comparisons as we did using OrthoFinder in the transmission experiment, but now we can look across experiments. And so here I'm just looking at diseased versus healthy individuals. So in our red ovals, we have, for example, on the furthest left in the dark red, this is from the ex situ transmission experiment. So diseased versus healthy differentially expressed genes. Then in the pink, we have our in situ intervention experiment. And then correspondingly, on our shades of blue, we have in situ in the light blue and ex situ in the dark blue. And what we find is that an overwhelming majority of the shared differentially expressed genes, of which there were just shy of 1,500, 2/3 of those once again matched direction. And as we saw in the previous experiment, about 1/3 of them had an inverse relationship. So once again, modulating some of these same pathways in different ways. However, when we then substitute in pairwise comparisons of our treated versus our diseased corals in our intervention experiment, and we compare that to diseased versus healthy corals in the transmission experiment, we see that the majority, almost 80% of differentially expressed genes have inverse relationships. This is telling us that those genes that were highly expressed when exposed to disease are far less expressed after they're treated. So we hypothesized from this that transcription patterns associated with disease exposure might actually be reversed following our antibiotic treatment. And this could be an attempt on the coral's part to reestablish homeostasis, to get back to normal. And I think it gives us a lot of hope that this antibiotic treatment is working the way it should. So across both experiments, we saw relatively clear patterns that SCTLD exposure causes strong changes at the gene expression level in both field and lab settings, and it's consistent with what we know about a coral conserved stress response. So generally, when corals are exposed to a multitude of stressors, they have immune responses that are remarkably similar. Now, from the intervention perspective, at the coral level, we know that intervention is working because it stops lesions from moving and killing the coral. At the transcriptomic level, it also appears that antibiotic treatment is effective because we're seeing a normalization of coral transcription following treatment. Now we've also seen, myself and others who are working in the field, that there's some survivors out in the field that might not even be treated at all, and it might be signs of SCTLD resistance. So for example, in this photo to the right, we have multiple Pseudoplorea strigosa colonies where you can see lesions on the majority of them, but the one in the top left does not have any lesions. So what is unique about that individual that makes it seemingly resistant to the disease? And as a follow-up, can we then identify traits that tell us something about SCTLD resistance, whether it be in corals in the wild or corals that we are actively managing? And so I want to spend the last few minutes to talk about two associated projects with this project that are trying to answer exactly this. The first is the SCTLD Resilience Research Consortium, or RRC, which was funded through the Florida Department of Environmental Protection. That project is wrapping up this year, in fact, this month. It's a project led by Brian Walker at Nova Southeastern University, but Josh is one of the members of that project team. And their main question is, why are some corals more susceptible to disease than others? And secondarily, can we then predict their resilience or resistance based on genetics? And so this team is a whole suite of different expertises at the ecology and molecular omics levels to look at potential indicators in Orbicella faveolata. And Josh's group at FAU Harvard branch is leading the coral genotyping efforts from that. And so what they found using a targeted sequencing approach called 2BRAD that looks at 10 to 20% of the coral genome, that genetic lineage alone is not a good predictor of disease susceptibility for this species. And so this whole experiment, they outplanted genotypes across Florida's coral reef, fate tracked them through time, and then looked at their susceptibility to disease. Then Josh's group did the sequencing on these to look at their genotypes and found that there were highly susceptible and highly resistant colonies found in several lineages. So there didn't seem to be any kind of clustering with related individuals being susceptible versus resistant, and found that some replicates within the same genotype had different levels of susceptibility. So from this, they suspect that 2BRAD might not be the best approach and that we might need either a wider approach looking at the entire genome or identifying individual genes that might still control or impact susceptibility to disease. Which leads into the second project funded through Florida's Department of Environmental Protection. This is a project that I'm a part of. It's SCTLD resistance and restoration. Our primary question is still the same. Why are some corals more susceptible to disease than others? And then our second question is, can we screen disease-resistant corals for targeted propagation and restoration? So can we take what we know about how these corals respond to disease, find the ones that are more resistant, and then frag them out so that we can propagate them, get more individuals, and eventually put them out onto the reef? So to do this, we assembled a great team of researchers between Mote Marine Lab, the University of Miami, and NOAA OML with ecology, restoration, and omics backgrounds. And we conducted the largest coral disease transmission experiment to date. It had 170 Orbicella faveolata genotypes with multiple individuals per, it was hundreds of replicates. An amazing effort done on behalf of Mote. And then we collected over 2,000 samples for multi-omics analyses. So when we look at the results of genotype susceptibility, we once again are finding that disease resistance is not just a binary response. It's not a yes, you're susceptible, no, you're resistant. Instead, it's more of a sliding scale. And so just like in Josh's field-based study, we found that some genotypes, they resist disease sometimes, but not always. So we find this sliding scale of susceptibility. Now, we're pairing this with high-resolution omics analyses. So we're looking at genotypes, we're looking at transcriptomics, we're looking at microbial communities to see if there are any common patterns occurring within our more resistant genotypes compared to our susceptible genotypes. And part of the project that I'm leading is doing whole genome sequencing. So instead of looking at a targeted part of the genome, we sequenced everything. And then on top of that, we are screening the rest of Mote's restoration genotypes to see if we can find those same patterns in other populations, so that if we find any evidence, genetic evidence of disease resistance, we can then screen for it across populations. So stay tuned for this project. It's actively ongoing, and I'm really excited to see the results that come out of it. With that, I've spoken for a very long time, and I see that we are right at 12:40. So I just wanted to acknowledge some co-authors from this study for which the paper is currently in review, as well as some individuals that were instrumental in allowing us to run both of these experiments, particularly when all of the labs were shut down due to COVID, and some of our sequencing facility colleagues that really helped us get this project off the ground and to help us optimize some of our coral sequencing needs. With that, I will take any questions. I'm happy to answer anything from the chat or Q&A. If you have additional questions that go beyond our seminar time here, our emails are listed here on the screen, and we'd be happy to answer via email as well. Thanks.
Dr. Studivan, thank you very much. That was an informative and exciting presentation. We don't have any questions just yet, but I'll start with a few. First, thank you very much for sharing your research and the information that you and Dr. Voss have kind of been collecting over the past few years. Like I said, no questions just yet, but I will start with one, give it a minute or two, and we can conclude. So I'm kind of wondering if there's been any interest in looking at the transcriptome of symbionts, although they could be considered they're very different species and types of organisms. I mean, do you know of any researchers at AOML or MOAT or FAU that might be looking into the difference gene expressions between symbionts and corals, or even the microbes or the microbiome of corals? Or do you think it's a little bit too differentiated to different organisms in order to compare transcriptomes? Care to comment, Mike? We might have lost him. I know that AOML is having some issues with their internet and power today. We'll give it a minute. Hopefully he can join back on. We do have one more question. Give it a minute. Hey, Mike, it looks like you're back. Hey, Mike, can you hear us? Hey, Mike, are you back online? You're unmuted. We can't hear you just yet.
Nicole, can you hear anything?
Yes. Yep, we can hear you now.
Okay, sorry about that. I'm not sure what happened, but AOML has had several power surges today, so hopefully I didn't drop off for too long.
No. So we have two questions. The first is, have anybody on your team looked at comparing transcriptomes of symbionts and their gene expression compared to corals, or is the differentiation between the organisms too big?
That's a really great question. So as part of my bioinformatics process, I actually do a very quick alignment to see what symbionts are in my transcriptomic data set, and then I align my samples to those symbiont references based on whoever's dominant, and I do look at symbiont gene expression. Symbionts are notoriously difficult to get good alignment out of. And so for the corals, where we might get something like 50 to 75% alignment of our sequences to the transcriptome, with our symbionts will get under 10%. And so I try not to rely too much on the data that comes out of that, just because I know it's really poor alignment. It's also historically been shown that symbionts might have really just generally muted gene expression, but that could also be the result of these bioinformatics challenges. So I suspect that as different transcriptomic references become available using better sequencing technologies, that this should improve. But right now we're just really limited in the kind of data analysis we can do at the symbiont level.
Thank you for that. Thank you for that. We have one more question for today. So the question is, or the comment is, great talk. Are there any plans to look at epigenetic methylation, e.g. methylation differences in the multi-omics study?
Oh, another great question. Let's see. None of the researchers in our group have epigenetic experience, and I'll be honest with you that it seems so far over my head, but it is definitely something that we need to start looking at. I do believe that there might be a subset of samples from the Restoration Consortium, the RRC project, that might be used for epigenetic, but it's definitely a budding field of research in the coral realm, where these techniques are just now starting to get adapted from, say, human biomed into working with corals. And so if there's a way that we can use already collected samples for that, I'm all ears, and I think that would be great.
Alrighty, with that, we don't have any more questions for today, but Dr. Studivan, thank you very much for your time today, and thank you all so much for joining us. Our next seminar is on May 17th at 12 p.m. Eastern Time, and our speaker will be Elise Larkin from NOAA Global Ocean Monitoring and Observing Network. Her talk is titled Biosensors of Environmental Change, Linking the Global Marine Microbiome with Ocean Processes. With that, the registration is up on our website. Thank you so much, Dr. Studivan, for your time today, and thank you everybody for joining.
Thanks everyone, and thank you Nicole for organizing.
Bye.